As food material continues its journey along the digestive system, it encounters new structures with new specializations. In order to understand the processes that go on here, it's first important to look at what these structures are. We recognize this as our stomach. This is the pyloric sphincter here. This is the first part of the small intestine. It's called the duodenum. Our consideration is only going to look at what goes on in the duodenum. The other parts of the small intestine way along here aren't part of this discussion at all. This is the gallbladder. The gallbladder is connected to the duodenum by the bile duct. This tube is the pancreatic duct. Pancreatic duct is connected to the pancreas, so it'll represent the pancreas out here. The liver is up here. So now that we've looked at what the anatomy is, we can consider what happens when the food materials get into this region. When we left the food material in the stomach, it was called acid chyme. pH of the acid chyme is around 2, quite acidic. The acid chyme is retained in the stomach by the pyloric sphincter, a muscle constriction right here. The stomach churns it around, and as it liquefies it, the more it churns, it liquefies it more, the pyloric sphincter can relax and let small volumes of it into the duodenum. The lumen, the inside of a tube is called the lumen. The lumen of the duodenum is quite small. It'll only accept small bits at a time, so maybe just a few drops get in here, and as it fills up, it actually puts pressure on the inside of the tube, and it has mechanoreceptors here. Mechanoreceptors, remember, they're nerve endings, and they'll send little messages back to the pyloric sphincter and cause it to constrict again. So as space becomes available, it releases the muscle constriction. Some comes in, it fills it up, it constricts it again. It actually will reduce the digestive activity of the stomach at the same time. This pH of 2 does some pretty different things going on in here. It affects chemoreceptors in the walls of the duodenum. Chemoreceptors, if we just look at the word, chemoreceptors are nerve endings, so that's the receptor part, that are sensitive to chemicals. The chemoreceptors here cause the release of 2 different kinds of hormones. One of these hormones is called CCK, and the other one is secretin. Secretin is released specifically because of the low pH. These 2 substances are hormones. Now, just like gastrin, which was released from the stomach, the hormones have to go into the blood, they travel around the body, and they affect different organs. CCK affects the gallbladder and the pancreas. Secretin affects the pancreas. let's consider first of all what the CCK is and what its effect on the gallbladder is. CCK is an abbreviation. It stands for cholecystokinin. This hormone affects the gallbladder and causes the release of bile. Bile is produced in the liver, stored in the gallbladder, and when acid chyme gets through here, the chemical nature, remember these are chemoreceptors, chemical nature of the acid chyme causes the release of this hormone, which causes the release of bile. Bile is an emulsifier. An emulsifier is something that breaks things up into smaller pieces. And we just have to return to the acid chyme and what the nature of the acid chyme is for a second. There's only two things that have had any digestive activity done to them in the acid chyme. In the mouth, some of the starch was broken down into maltose. In the stomach, some of the protein is broken down into polypeptides. I'm going to need a little space here, just a minute. We're considering what bile does. Bile is released from the gallbladder. It's an emulsifier. One of the components of the acid chyme is actually the lipids. Lipids don't mix well with our body. We have a water-based system, and lipids don't mix with water. So what happens here, if we have a lump of fat, for example, is the bile will treat that lump and break it up into tiny pieces. That's the process of emulsification. It's a very important physical digestive step because emulsification takes this lump of stuff, this larger piece, and breaks it into smaller pieces. It would be known as fat droplets, for example. The fat droplets have got a tremendous amount of surface area compared to what they started out as. And more surface area allows enzymes more contact, you get more enzyme contact, and you can have a more efficient digestive process. So this is a very important step in the digestion of fats. They first have to be emulsified, the digestion comes later. CCK and secretin also affect the pancreas. And cause the pancreas to release pancreatic juice. The pancreatic juice is a complex juice. It has 6 components. We just list those 6 components here. Water. Water is required throughout the digestive system because digestion is a hydrolytic process. All of these molecules have to be taken apart. Every time a chemical bond is broken in them, a molecule of water is added to it. So it's hydrolytic reactions. There's bicarbonate ions. Actually, they come as sodium bicarbonate, but it ionizes. Bicarbonate ions like that. The significance of the bicarbonate ions cannot be understated because we have a substance which has a very, very low pH. We have acid chyme is a pH of 2. The enzymes which work in the duodenum don't work at a pH of 2. They work at a pH of about 8.3. So what the bicarbonate ions do is they raise the pH to about 8.3. And they buffer it, keep it at 8.3, because the other 4 components of pancreatic juice are enzymes. And they all work at 8.3. So we've got 6 components: water, bicarbonate, and 4 enzymes, 4 types of enzymes. We have to look at what each of those kinds of enzymes are and what each of them do. So we're going to return up here. I can leave that. Our topic of 4 enzymes. Let's consider the lipid digesting one first. Its name is lipase. And what lipase actually does is it takes the fat droplets with all the surface area, they're lipids, and it digests them into fatty acids and glycerol. The second enzyme that's in pancreatic juice is trypsinogen. Trypsinogen, if you recall the word zymogen, trypsinogen is inactive. It's a protein digesting enzyme, and it can't be produced in an active form. It's produced in the form of trypsinogen, and it's actually treated by an enzyme and converted into trypsin. Trypsin is the active form of trypsinogen. The enzyme's name is enterokinase. Just to make a connection for you, pepsinogen and trypsinogen are both zymogens. Pepsin and trypsin both end in -in. Most enzymes end in -ase. These 2 enzymes are called protease enzymes. Pepsin and trypsin are protease enzymes. So there's the -ase ending. These are active enzymes. Trypsin takes the polypeptides, which are a component of the acid chyme, and it breaks them down into smaller pieces that are just called peptides. The peptides could be a single amino acid, or they could be dipeptides or tripeptides. There's various lengths. It all depends on which peptide bonds, the peptide bond between the amino acid, which ones get broken. So you get various lengths of those peptides. We've seen an amylase enzyme before. We saw it in the mouth. Salivary amylase digests starch and converts it into disaccharides. The most common one is maltose. So we have starch, which would be maltose, and that's a disaccharide. Our amylase here, pancreatic amylase. The 4th enzyme works on the 4th type of food particle. We've got lipids, we've got our protein component, we've got our carbohydrate component, we also have our nucleic acid component. Could be DNA or RNA. We don't think we eat a lot of DNA and RNA, but every time we eat something that is cellular, we're also eating the chromosomes that are in the nucleus, that sort of thing. So we do consume DNA and RNA. It's not a big part of our diet, but it's there. Nucleic acids are broken down into nucleotides. By nuclease enzymes. I should have written this differently. I've got the enzyme, I've got the substrate here. Let me correct that for us. The enzyme, nuclease, it takes the nucleic acids and converts them into nucleotides. So that's our nuclease enzyme. We've got one more step to consider in the small intestine, the duodenum, because these things aren't ready to be absorbed yet. They're not, digestion isn't finished. It's finished here. Fatty acids and glycerol, those are the unit molecules. So that's okay. Peptides, maybe there could be some amino acids in there, but they're dipeptides, tripeptides. These are still too big to be absorbed. Maltose, disaccharides, too big to be absorbed. We do eat other disaccharides. Sucrose, for example, is a disaccharide, and it has to be broken apart as well. Nucleotides are still too big to be absorbed. This one's okay. So there has to be another set of processes that goes on in the duodenum to get these things small enough so that they can be absorbed. That other set of processes is not a result of pancreatic juice. The small intestine actually secretes its own digestive enzymes. So I have to get some space here. I don't need any of this. The duodenum secretes its own enzymes. We've got three types of food particles that aren't completely digested yet. The duodenum secretes intestinal juice. The three kinds of food particles that aren't completely digested yet, we've got the protein component. The protein component are peptides. We've got our carbohydrate component of the food. They're all disaccharides, for example, maltose. But there are other disaccharides in our food. We also eat sucrose, lactose. Those are disaccharides as well. Sucrose, lactose, they get all the way through the digestive system. They're not digested at all until they encounter disaccharidases in the intestinal juice. The other component of food that isn't completely digested yet are the nucleotides. Intestinal juice contains peptidases, which break these down into amino acids. Intestinal juice contains the enzyme that's going to break down disaccharides. It's simply known as disaccaridases. So if we're dealing with maltose, for example, that would be maltase. When this is all done, these are all monosaccharides then. So for example, with the maltase example here, we would have glucose. The nucleotides are broken apart by nucleotidases. And the components here, we have individual bases, we have sugars, and we have phosphatidyls. So there we have it. The components we have from intestinal juice, 3 of them. One right here, one right here, and one right here. The products of all of this, amino acids, monosaccharides, bases, sugars, phosphatidyls. Don't forget, we've already got the fatty acids and glycerol from the lipase enzyme. All of these things, by the time they're moving along through the small intestine, they get out of the duodenum, all of these things are ready for absorption. The topic of absorption is a big one in itself. It comes up in a further video. Thanks again.